Yeah, record and transcribe. Okay.
Go ahead. It is just started.
Okay. Okay. So, well, hi everyone. I hope everybody like everyone who is interested has logged in. So today we are going to have a second lecture in the online series, and we have Dr. Ram Pradeep Samyappan today to share his experience. So I welcome him, and welcome everyone who have already talked, whoever have logged in today. So Dr. Samyappan has worked with Professor Hans Hebert and Professor Caroline Jägershold in Karolinska Institute, where he has worked with trying to visualize the exosomal microRNAs, and he has also worked with membrane proteins, how it integrates in the nanodisk. So he has quite a bit of experience of working with different kinds of samples, and currently he's working with Thermo Fisher. So he will share his knowledge about handling various kinds of samples in negative stain as well as in native condition. So we are going to have a very practical talk today about how should you analyze your sample before you start to collect data for high resolution cryo-EM. So I would request him to take us through his talk, unless someone else has something else to add to it before he starts to talk. Like, I mean, if Professor Pradeep wants to talk something about it.
No, nothing. Let's hear him, and then
okay.
And after that we will have questions. Yeah, thank you.
Okay. Okay, so yeah.
Can you please present the screen and then get going?
Yeah, Professor, I'm sorry, Dr. Samyappan, please go ahead with your talk.
Sorry for the interruption. I think Pradeep is having some issue with his internet. He will be joining soon.
But he has already logged in, Professor Pradeep, right?
Yeah, I think there is a. Sorry, I mean Ram Pradeep is.
Okay.
This is Pradeep Samyappan. Yeah. So
yeah,
this
Pradeep.
is yeah.
Sorry.
So let us probably wait. Where is he joining from?
So he's joining from Bangalore actually. So he's just moved into Bangalore, so he has been like having some issues with setting up the internet.
Now he's logged in.
Now he's
logged in. Okay. So let's have a talk now. Okay.
He's he's coming in.
So let's get going.
Sorry for the interruption. There is something.
No, no issues. Well, these are part and parcel of this. So, yeah.
So can you see my screen now?
No, it's blank actually.
Your network is poor as well, so maybe that is another issue.
How about now?
Not yet.
Not yet. Yeah.
Maybe we can.
Now it is.
Maybe we can. All camera can be switched off, so that makes it easier.
Just let me know once.
Not yet actually.
It's visible.
Okay. No, it's not visible.
I think.
Okay. Sure.
Ram, if you don't mind, can you just again log off and log in so that it's visible?
Sure.
Yeah.
Hello. Sorry. அப்படியா
You are visible.
And
Oh.
on the screen.
Okay. Oh, CVCMD, yeah.
Screen is still not
Ram, you
visible.
can switch off your video. Maybe you can switch off your video and then show the slides.
No, his network is poor. That is the situation. Something has to be done about it. Because all these things will have some
Okay.
amount of videos and all that. It will make it difficult.
Sorry,
Um,
Ritesh, I'll be back in a minute.
Yeah, I mean, that's okay. That's okay.
so Ram is trying to share the slides with me, so I can I can project the slides. Maybe that way it will be a little bit easier for everyone. Just
Okay.
give us a couple of
Okay.
minutes more. That's it.
Yeah.
Thank you. I think Ram's network is quite slow.
Okay, so how do we resolve this issue then?
Ram, you are on mute.
How about now?
Just give a couple of some
Can
10
you see the
seconds.
screen
Maybe
now?
there is a delay. We can see your screen.
No, I can't actually.
You can, or
No, we can't. Your
No,
screen is not visible.
Yeah, it's blank actually. It's white.
well,
mean, I
Kumar,
could
I have
see-
also sent it.
I think your network is too slow to upload the slides on my in the Teams as well, even if you have to send it to the Outlook. If you just get the link, I think I can do that.
No, it is not only that, his voice also will be very slow. It will not coordinate. Dr. Samyapan, this is
Sir,
going
could
to
you
be
please give us?
even if
Could
the
you please give us 5 minutes? We'll sort out the issue.
Actually, your voice is
we'll
also
come back.
cracking a little bit. Yeah. Okay. Okay.
if we can connect to another network, maybe another phone line or something, maybe better. Well, so for everyone, we will wait for another 5 minutes. Dr. Ritesh, so I'll be 11:16. So I'll wait for another 5 minutes, and then if things work out, we will proceed. If it doesn't, then we will have to reschedule it.
Okay. Okay. Sure.
So many people.
Yeah, sure, sir. Give us five more minutes, and
Sure, yeah.
by 11:20 we will be back. Yeah.
Sure. Yeah. Hello, Kumar.
Hello.
Yeah, hello.
Very sorry. I think now I will send you a new link. This is a Google Hangout, and I tried with Kumar quickly, and I tried to screen share and talk as well. It works, but let's try this. If it doesn't work, probably we can try to reschedule it. Let's give it a last try, probably.
Okay.
Ram,
let me
before we
try.
switch back.
So this is the link, and I will quickly log into that one.
Ram, just before this, can we try one last time to share the screen in this Teams itself? Can you switch off the video and try to share the?
Yeah, now we can see it. Can see your screen. Okay.
How about now?
Yeah, we can see it. Is everyone able to see it, or?
Yes,
it's fine. Yeah.
Okay.
Go ahead.
Okay then. Yeah, let's
And my
go ahead.
voice is yes.
Yeah. Please.
Yeah, firstly, I would like to ask my sincere apologies for this technical glitch. It was something unexpected and unintentional, and sorry for wasting your time. Actually, I'll quickly jump on to my presentation. This is Ram Pradeep Samyapan. I'm the application manager and an application scientist in EM services, working at Thermo Fisher Scientific. Before, I'll quickly share my background. I worked in a company called Veranova AB in Stockholm, Sweden, as a senior scientist. I have expertise in the field of structural biology, especially in cryo-electron microscopy and negative staining. And I work with different particle types, virus, lipid nanoparticles, liposomes, and different drug delivery particle types. And also worked in BSL-1 and BSL-2 laboratory, adhering to the GMP guidelines. And previously, my research was under Professor Hans Sebert Group at Karolinska Institute. I worked on membrane protein structures, especially cryo-EM as an important method for determining the structural information for sodium potassium pump protein. And with that, I would like to start my presentation. So in the next few minutes of my presentation, I will try to cover information regarding transmission electron microscopy and main significant components of TEM electron microscope. At least the first 10 slides will be the introduction to EM, considering the general audience over here. And then later I will introduce to two methods, negative staining TEM and cryo-electron microscopy, advantages and limitations of the two methods, and how cryo-EM plays an important role in determining the structures of several biological samples at both structural and functional level. And also a few technical things involving few techniques related to sample preparation and also sample optimization procedures, and how sample preparation plays a critical role in cryo-electron microscopy. And later I will go through the single particle analysis workflow, right from the start until the data collection, and then how application of electron microscopy works in different fields, be it pharmaceutical sector or else academic sector. And I will also touch upon principles of tomography a bit, and a few insights about the tomography process, how it is done, and what are the equipments that are used for the tomography process, but not in depth. and future perspectives, how both cryo-electron microscopy, how both single particle analysis and tomography plays an important role over here. And please interrupt me in the middle or at the end of the presentation if you have any questions, or we can take that later. A short introduction about the microscopes and microscopy. Previously, during our schooling, we used optical microscope, and mainly it has a basic lenses, and the magnification is too small. And we have done several experiments to examine the cells and tissue samples. But now the difference between the optical and electron microscope is, yes, obviously from the name, it shoots electrons from the electron gun. And then we have a specimen with different contrast. And of course, we can achieve huge magnification, and we can also try to solve atomic level crystal structures of biological samples, especially proteins. Since I have worked on proteins and membrane proteins, it is a very tricky and complicated process, but latest sophisticated equipments are can help us to achieve that atomic structure. So I'd like to show a basic idea of what is STEM and how does it look like. Of course, for anything to be projected, we need a source. For example, if you take our planet as an example, our source is sun. The light from the sun generated, and that's how we get light. Similarly, if you take electron microscope, there is a source which is electron gun, and then there is condenser lenses, C1 and C2. I will come back to that, what is C1 and C2 in detail. And there is a specimen of interest. So whatever specimen is placed, and there is a specimen of interest, and objective lens, where we can try to develop a very good contrast, and apertures of different sizes. For example, we have apertures right from 20 to 100. The size range is basically to eliminate good and bad electrons, for example, and to give a very good contrast. And this in particular, the aperture sizes are more important for biological samples as well, to get a very good contrast, and also the low electron dose should be there. And finally, we have detectors. Whatever we see in the specimen should be able to visualize in the fluorescent scheme with the help of the microscope. Sources, lens, and the detector are all the important components of the equipment. As I said here, I will go through the condenser aperture. This is the electron gun, and as you know, electron gun is the most important component, and we have different filaments. Example, lab-6 and tungsten filament. And over here, it's a condenser aperture, C1 and C2. Basically, it has to play around with the spot size. Spot size starts from 1 to 11. Lowering the spot size, meaning the intensity is low. The higher the spot size, the intensity will be higher. And over here, there will be your specimen of interest, where you load the sample. And later, it's an objective aperture and different apertures. Try to see if we can try to eliminate the intensity. And finally, there is an image record system where we have different detectors right now. As I said about filament, lab-6 is much more stable, and tungsten filaments, we used it historically. As I said before, this is about the condenser lenses and objective lenses to maintain the focal strength, and also the projector which magnifies the image, and the images will be displayed on the CCD screen. This is something important topic I would like to touch upon actually. So the compute stage plays an important role over here. Once the sample is inserted, the sample should be in focus. The image should stay in focus, even if the specimen is tilted. For this, what we do is there is a process called U-centric height adjustment. I'll come to that, but the main idea behind it is to see if the sample lies in a correct focal plane, so that we can visualize the image in a correct focus. There are different holders right now: single tilt holder, double tilt holder, multi-tilt holder. So irrespective of irrespective of the usage. If we have many particle types, we can use multi-tilt holder, and if we have a single particle type, we can use single tilt holder. So there are different holders right now. Now the latest one is ELSA cryo holder. As I said about the detectors, right now, previously we don't have many detectors, but right now we have latest advanced types. So cryo-EM as a whole, there was a resolution race in 2000, since 2007 or even before that. And before 2000, probably X-ray crystallography was played an important role in determining crystal structures of biological samples. But right now, slowly, after 2007, cryo-EM took a advanced step. And this is after 2017, once Jacques, Richard Henderson, and Joachim Frank's group, once they received the Nobel Prize on cryo-electron microscopy, then we are kind of aware of the cryo-electron microscopy as a technique as a whole, and also the sample preparation and sample optimization techniques behind it, and how we can try to solve the crystal structure, how we can try to solve the structures with cryo-electron microscopy. And people slowly transitioned from crystallography to cryo-electron microscopy. As you can see, this chart explains what cryo-electron microscopy can do and what are the limitations of NMR and X-ray crystallography. As I mentioned before, briefly, the revolution started in 2007. Previously, there was microscopes from different competitors as well, but the detectors were not that good to achieve a high resolution. But once in 2012, or if I remember correctly, in 2012, after the Titan Cryos launch, there was a very good research articles with high atomic resolution, and which you can able to achieve with advanced detectors as well, and in a very quick time frame. As you can see, what cryo-electron microscopy can do. Apopheratin samples, it's a main control sample to determine the efficiency of any cryo-electron microscopes, and where we can see that there was an 1.62 angstrom resolution already. And right now, as I speak now, and there is an article for 1.2 angstrom resolution as well. So people are trying to make use of the equipment and to get a better resolution and better structures as well. And on the right side, you can also see the single particle analysis of virus sample. It's around 1.56 angstrom. So getting into the insights. So right now, we have gone through what is electron microscopy, what is transmission electron microscope and its components, and how cryo-EM plays an important role. So with this insights, let's go to the TEM techniques in detail. So briefly, we have two methods: negative staining, NSTEM method, and cryo-electron microscopy. As a beginner, if I would like to visualize my sample of interest, I would like to go with NSTEM technique, negative staining. I will tell you what is that and how quick is that. So basically, what you need is 3 microliter of sample. That's it, 3 microliter of the sample, but concentration plays a very important role here. So be it a liquid form, or else if it is suspended in a liquid form. So we need to have an optimal amount of concentration. I can tell an approximate amount of different samples, but that's a rough estimate. We have to identify the concentration level for our sample of interest. So this 3 microliter, and it is placed on the grid, and the sample is applied on the grid. There is a waiting time period for approximately 1 minute once the sample is incubated on the grid. And afterwards, there is a washing step. there is 3 washing steps, basically to get rid of the debris. And if there is any buffer components or if there is any huge aggregates that can be washed away, retaining your sample on the grid. And after that, there is a staining process. So normally we use uranyl acetate, uranyl format, or PTA, phosphotungstic acid. Over here, the concentration of all the staining agents plays an important role. So at least 2% uranyl acetate or else 1% is very important here. If it's an excess, then you will get thicker stain. Ultimately, you won't see anything on the grid. So the staining process is very important, and how quick you are. So it has to be approximately 30 seconds, 45 seconds, and up to 1 minute. So more than 1 minute, it might give you a dense thickness. Okay. So when it comes to NSTEM process, so what are the things that we can visualize? So if you prepare your particle of interest, let's say protein samples or virus samples.
Ram, you are on mute.
Ram, you're on mute.
Now?
Yeah, now yours. Yeah, please go ahead.
So after this staining process, you assume that we have prepared the samples on the grid, and the next step is to visualize the grid in the microscope. So what is our aim over here? What is the objective?
Sorry, you said it is a 3 microliter sample, and after that, I did not hear.
Okay. I'll come to that. TEM techniques, we have 2 methods, NSTEM and cryo-TEM. What's important over here is your sample of interest. Be it in a liquid form or be it in a suspended to a liquid form. 3 microliter is more than enough actually. But what's important over here is concentration of the sample. And concentration varies quite a lot between NSTEM and cryo-TEM. But based on my previous expertise, I can tell you the approximate range for virus or protein samples. But it's very important that we should know what concentration is optimal for NSTEM. It has to, yes, keeping that in mind. The sample is incubated on the grid, and the incubation time is 1 minute. And after that, there is a prior washing steps. The washing step is mainly to remove the debris from your sample preparation. And once that is done, there is a staining process. So what we usually normally use in the market is uranyl acetate, uranyl format, and PTA, phosphotungstic acid. Over here, the concentration plays an important role for the stains as well. We need at least 2% or else 1% in terms of the concentration. The excess the stain concentration will result in excess thickness of the grid. And ultimately, you might not see anything in the sample. You might not see anything in the grid once you are in the microscope room. So the uniform sample uniformity is very important, and sample cleaning and then staining, sample staining. So uniform staining is also very important over here. So what we can quickly see in negative stain is there should be some objective. The objective is to visually see the overall morphology of the sample. So whatever sample, we can see if the sample is spherical in shape or circular in shape, whatever it is. And we can also see aggregation mainly. So if there is any clusters, we can try to see sample of interest is a kind of aggregator, or it tends to aggregate. So you can go back to the preparation and you can work on optimizing your protocol. And then later what you can see is particle size distribution. You can do that based on, for example, if you take adenovirus vector, we can try to do particle size distribution based on size and also based on the density filled in. So if there is any, if there is anything, for example, if there is any drugs that is encapsulated into the adenovirus vector, you can see that with the negative strain, the particles will be dense, and if it's broken, the particles will be. You can see that it's broken particle. So you can try to classify as filled and empty. And based on the visualization, you can also judge do purity assessment and sample integrity. Yes. So all these steps you can do it in a very quick manner. I would say if you have a sample ready, you can get the results within a day, within half a day, for example. And you can try to do at least 3 particle types or 3 samples in array, 3 or even more actually. So it's that quick it is, and you will get a basic idea of how your samples looks like. The next step is cryo-EM. Cryo-EM, again, here the concentration plays an important role. As I said, for NSGEM, you need at least 10 times lesser 10 times lesser concentration than cryo. So if it is 0.5 mg per ml for cryo-EM, I would like to have 0.005 or even 0.001 mg per ml for NSGEM, at least 10 times or 20 times. For cryo-electron microscopy, the most important part is, of course, after once the sample is ready, vitrification procedure. So your sample will be placed in a chamber. So either it's a Vitrobot equipment or it's a Leica equipment in a temperature controlled chamber and also humidity control. Normally, either we can try to freeze in 4 degrees Celsius.
Are you moving the slides, or you're still on the same slide?
On the same slide, sir. I just want to speak about the method and then I can go. Yes, so over here it's the vitrification process where you need the temperature and humidity plays an important role. And once that is done, it is plunge freezed in ethane. And then the grids will go to the microscope. So what you will see is your particle will be in the native state. And these are the objectives that you will be studying in cryo-electron microscopy. And this is how the NSGEM workflow will be. So once you have the sample on the grid, its glow discharged to make the grid hydrophilic. And then your sample of interest is applied. Then there is a step called water wash. After it might be either 1 time or 3 times. And afterwards it's staining. The sample is embedded on the stain and it's getting dried. There is a drying time for approximately 5 to 15 minutes, and then it's stored in the storage box for later visualization in the microscope. So this is the whole NSGEM workflow. And I said about grid multiple times. So what is grid and why it is used in microscopy? So normally we use copper coated grid for biological samples and also gold for standard samples as well. And it is available in different measures. Measures meaning it's different size holes actually here. And this is more important for biological samples. At least you need 200 to 300 measures so that you can see that the sample dispersion is very clear. And on top of the grid, there is a faint material that is coated, which is called mainly a carbon film, or else it can be foamware and also silicon monoxide as well. But mainly we use carbon and foamware. And there are 2 types. So it's wholly carbon film for single particle analysis, which is very frequently used. Hydrophilization of the support. So for example, the grid from the manufacturers, they might have a debris on their own. So what's important is there is a process called glow discharging. And for example, for the aqueous solutions, the grids in nature, by default, it would be a little bit hydrophobic. So what we do is we do a glow discharging process to make the surface of the grid hydrophilic. And the procedure is by inducing a current in a stable tight vacuum. And then the ionization chamber does the rest. And once this process is done, this process usually takes, I would say, 5 minutes. Once this is done, the grid is ready to be prepared. Preparation protocol. So yes, this is how you have to hold the grid. The sample is dispersed, and either you can swipe the liquid with the help of a filter paper on the sideways or else on the bottom part of the grid. But make sure that you won't bend the grid. That's very important. And then the next step is washing steps. Similarly, with the filter paper, gently swipe around the rest of the liquid. And the final part is the staining process. And the grids are ready for imaging. Now we can see a few examples, and this is from my own example actually. I worked on a membrane scaffold protein. It's a nanodisk actually. It will form a belt-like structure. Upon visualization and negative staining, PTA, it will form as a stacks like this of different sizes. So what you are seeing is a negative chain of empty nanodiscs of different sizes, and approximately it is 17 nanometer in size. And these are the side view of the discs. And with the help of uranyl acetate, you can see the top view as well very clearly. So this is the reference example from uranyl acetate. And I also tried uranyl format, but it didn't work well. But with uranyl acetate, you can see the particle features very clearly. But with the negative stain, the limitation lies on the resolution. So you shouldn't take this as a final data. What you have to do is you have to take this as a rough initial model. So you can try to do the image processing and get an idea of how your sample looks like. And later you can move on to cryo. So this is a kind of, I would say this is a kind of optimization process. And with negative staining, there are different particle types, as I said, adenovirus, ISCOM matrix samples. As you can see, there are aggregates, and then also adeno-associated virus, where you can classify, try to classify empty, broken, and also some doublet assemblies as well. As I said, it's there is a desalting process that depends on how well the sample looks like. If it needed, it can also be done, and to purify it. It's mainly the purification protocol. With the negative staining, you can also see the sample aggregation. And quickly you can judge that your sample is aggregated so that you can go back and see how you can optimize your protocol. And this is AAV's examples. And the main advantage in NSTEM is you can quickly go through screen the samples, and you can quickly judge how your samples look like. So the the judgment of your how the samples looks like, it is a very quick and convenient process, I would say. With this aspect, then we will move on to cryo electron microscopy. So now we know what is negative stain and how quickly we can screen and get results from negative stain. Now it's time to move to cryo-EM data. So the main objective is we can try to answer biological questions, and we can also do biochemistry optimization. And with the sophisticated equipments right now available in the market, there is no need for microscopy expertise. So anyone can quickly learn the technique and quickly can operate the microscope and try to go through the data collection process. But what's important is your sample optimization and sample preparation. And that comes to the research. So your scientific knowledge plays a very important role in preparing the samples. And once you get a very good sample, then you have equipments that can take care of the rest. But the background and backbone lies in the sample of interest. The sample should be in a very good quality. Let me go through cryo-EM workflow. As I said, it has to be glow discharged for biological samples. And once it is glow discharged, then it is the grids will be placed in a plunge freezer. This is a Vitrobot equipment, and your sample of interest will be either placed on the side base, either on one side or two side plotting. So you can try to optimize it, optimize the protocol. And it's a humidity control chamber where you can play around with humidity. Maximum it's 100% or 19%, and also temperature controlled. Usually for virus samples, 16 degrees works fine. For protein samples, probably 4 to 6 degrees works fine. And it is once the vitrification process is over, then the sample is stored in the grid box. And if it is shipped, then it's a cryogenic container, and it's quickly transferred to the cryospecimen holder, then to the microscope for the data collection. Yeah, this is how the sample preparation process, the vitrification process at least looks like. The sample is incubated on the grid, and it is blotted with the filter paper, quick freezed in liquid ethane. The freezing should be really quick in 1 or 2 seconds. So if you take excess time over here, then you might see ice contamination in the samples, in the images. I will show a video quickly how the entire workflow looks like. Please let me know if you couldn't see the video. Okay. This is exactly a representation of what I have explained, what I tried to explain. How quickly you can prepare the samples, and when it comes to data collection, and also the final 3D reconstruction with different image processing softwares. And right now, with the data collection goes really quick. Within few days, you can get a result with a very good quality sample. So I'll share you a few examples of how cryo-EM particle types looks like and how NSTEM looks like, so how negative stain samples looks like. So this is as you can see, this is a stain. This is from the strain NSTEM of adenovirus, and this is from the cryo-EM. The density comes from the ice. The right side, you can see it's a cryo-EM sample of, I think this is a AAV virus, where you can see doublets and triplets very clearly. And also you can see the density filled and empty very clearly. And this is NSTEM's NSTEM representation. So I have gone through both the methods, NSTEM and cryo-EM, and there are advantages and there are disadvantages in both the techniques. I will also focus on the limitations as well, which is very important here. So of course, in cryo-EM, the main advantage is you can see your specimen of interest in a native state. And there is a clear discrimination between filled and empty. If you take one particle type as an example, if you take liposomes, you can also try to see the membrane thickness at a higher magnifications. So what you can see is the sample of interest will be almost close to the native state. NSTEM, it's a straightforward methodology, but with the staining agent that comes in practice, probably the resolution is very limited. It's too limited. So you have to just take NSTEM as a reference method. And debris and background material can be easily identified. And as I said, resolution is limited, yes. When it comes to limitations, it requires a tedious preparation, yes. Right from the sample preparation and concentration optimization, this is a very important role. Sometimes you see a lot of samples on the grid, and it will be too crowded, and which is very difficult, very complicated for image processing. So we have to make sure that your concentration is optimized, and you can see the uniform distribution of particles like this. And then it has a necessity to work in low electron dose. The reason being, for the biological samples, if you apply high electron dose, you can see that the particle tend to burn very quickly on the grids. And over here, NSTEM specimen preparation with the stains, of course, it can affect the particle morphology as well. And the data will be the initial model will be as a reference. So you use that as a reference and try to put this as a reference model for the cryo-electron microscopy. And in the NSTEM, when it comes to contrast, we can get a very good contrast. But on the other hand, in the cryo-EM, the contrast is a little bit weaker because there is no staining agent. Whatever you see with the sample is frozen in liquid ethane, and then whatever you see is based on the ice thickness. And with these two techniques and with the cryo-EM data, what you can try to achieve. So it can handle the complexity of different infectious diseases by you can visualize multiple conformations, for example, and try to see the interaction between protein to protein complexes. And you can also see how the post-translational modification works. And what you need is some microliter sample. It's as I said, it's approximately 3 microliter. That's it, and no crystallization process is needed. So this is the total single particle analysis workflow. The first step is the sample purification process, which plays a vital role here. And the next comes to quality control, where you can try to screen your sample of interest and see, and you can get an approximate idea of how the sample looks like. If the sample doesn't look good, then go back to the previous process and try to optimize and come back here. And once it is optimized, and once you can collect at least a preliminary data, let's say a few thousand particles, then the next step is to go to the vitrification. Then the next step is to go for cryos with a high-end data collection, where you can go through all the process and try to do the data analysis later on. And for example, if the samples are prepared and if you want to store it for a longer time, of course, you can use it. You can store it in liquid nitrogen reverse, and it has a storage capacity. At least, I would say, at least for the first 14 days, your sample will be really good. But the longer incubation time on the reverse, probably it might cause ice contamination as well. I would say shorter, try to image your sample as quick as possible in order to avoid other external factors issues. And this is how the cryo-EM workflow at Thermo Fisher looks like. So we have different equipments for different purposes, but I will just quickly go through this slide. So we have for the vitrification process, we have Vitrobot of different Macs. And there is a screening equipment for the glaciers where you can try to screen the particle types. And you can also try to get a good resolution here. But ultimately, with the cryos and with the high-end detectors, you can get a very good resolution later on. And for tomography, it's a totally different model. But of course, we have equipments to face all kinds of issues. And this, as I said before, I will quickly go through the image processing protocol. And this is the upper ferritin sample as a control sample. Initially, we had 350 micrographs. The next step is particle picking process, around 50,000 particles, but that will filter out after once you remove the noise. Each stage, 2D classification, 3D classification, you will be filtering out the particles. Ultimately, you will get very few particles, but the resolution will be really good. But the reason why I want to show is the process of image processing. The steps of image processing looks like this, irrespective of whatever software you use, eMan or Relion or CryoSpark, whatever you, the principle behind it lies, it's the same. And yes, right now you can get very good resolution data with different particle types. And here are a few examples: 1.15 angstrom upper ferritin and 10 megadalton protein for poliovirus. Sorry, 10 megadalton size poliovirus samples. And there is also a very smallest protein example. It's 40 kilodaltons actually. And this is to see how quickly cryo-EM was used for the image processing process. Was used to collect atomic resolution. We can take our COVID pandemic as a very good example. Actually, it started approximately in 2020, and within 18 months' time, what we got is data, cryo-EM data of the COVID sample, and also the drug delivery drug targets, actually, especially pharma companies. They used lipid nanoparticles as an example, and also liposomes, and they worked on different particle types, but ultimately the overall crash, the overall period was very quick. And I think it took 18 months for most of the companies to come up with the drugs. And cryo-EM played a very important role over here. GPCRs, as you know, GPCR, G protein coupled receptors, has a huge pharmaceutical significance, and it's highly used. And these are the reference articles, but you can read. There are lots of articles over here attached with GPCRs with different resolutions. And few virus samples as an example. This is GABA receptor protein, membrane protein. So I would like to put a membrane protein. It's a 3.4 angstrom resolution. It's very good. But you can see the images. It's really complicated. The complicated part lies in the preparation actually, and to see it and to get a 3.4 angstrom resolution is really a huge step actually. Apophoratin, I have already mentioned about it. And AAVs we can see very quickly, very clearly, and the optimization process is quite easy. At least I would say I know the concentration roughly which one works fine. So AVs are very good. And also in terms of classifying full and empty particles, it's cryo-EM plays a very important role over there. So if you see a density, it can be classified as full. If you see a density similar to the background of the grid, so you can classify it as empty. So if it's a density, it's full, and if it's not, it's empty. So that's quick it is. And you can also see doublets and triplets occasionally. And I would like to say this one. So this is the ice contamination. So this is not a particle. So you can see ice contamination here and there. That might come during the vitrification process. As I said about the classification, empty and full. And liposomes. So liposomes, lipid nanoparticles, and right now the latest one is exosomes. So all these plays a very important role. And with the help of cryo-EM, there is a lot of pharmaceutical significance attached to it. For example, exosomes, there is a lot of cancer research studies going on. And with the high resolution images, you can also try to measure the membrane thickness. And if there is any drugs infused in it, you can also see the internal structures very clearly. But otherwise, based on the particles, you can try to classify as unilamellar or multilamellar, or else try to measure the size distribution is also possible. So now, speaking about the challenges and the important parameters. So this is after the grid after the sample is prepared. So what's important over here is screening process. And right now, EPU, the software can do this automatically, but the important parameters to know is this is the area of the grid as an example, and you can see these are the areas that is too dense, the ice is too thick, and over here you can see there is different gradient of ice, where is thin and thick ice. I will come to that in the next slides. So this is how you have to choose the grid. So grid should be not too thick or not too thin. A fuzzy area like this will be too good for the samples. And if you take protein samples, so you can see in a thin ice area, there is no samples at all, or else very few. But most of the protein samples lies close to the carbon film. So it's better to use the holy carbon grid for this purpose. And it's better if you can avoid thick area, but if you visualize thick area, you will see a lot of particles, but it is limited to resolution. So you can try to pick particles here, but the resolution will be limited because of the contrast. And this is how this is just an example to see difference in ice thickness. Yes. So what we can do is we can try to optimize our preparation by visually seeing your particle of interest in the microscope. And once it is optimized, you can try to acquire good quality images, and later, yeah, we can try to answer our biological questions. So what's it? Next step is I will try to go through the tomography process. So the first part, single particle analysis is done, and the next step will be tomography. The principle lies behind the tomography is same, but what's important over here is you can once the sample is inserted, you can try to visualize the sample of once the sample is vitrified, you can try to see your sample of interest in different angles. And as you can see, recently the cryo-ET is also picked up over SPA. And I think over a period of time, micro-ED is also coming up. So there is a lot of other methods booming up. But irrespective of the techniques, the principle behind it is same. So we need to answer a biological question irrespective of whatever technique we use. This is the common workflow of the tomography process, and we use Amira software, which is an in-house software for classifying the particles. And this is the time scale that we can expect to see the results actually. So it might take initially, even before that, there is a lot of research work. Time-consuming process will be in the sample optimization. Once that is done, then it might take days or even months actually. And over here, the cells are, you can visualize tissue samples, for example, but over here, the important thing is it's not fixed or it's not stained. Your sample is preserved in a native state during the vitrification process, and you might get a higher resolution. And you can see the information very clearly, actually. I will share one video, how the tomography process looks like. Over here, there is an example of a virus in solution, and it's vitrified now at a temperature minus 180 degrees. You can try to take around the different tilt angles. So what you can see is your sample of interest will with different orientation, for example. And more cancer-related research is ongoing, and people tend to try to use cryo-electron tomography for this purpose. And this is one reference I would like to show, a result achieved from cryo-electron tomography. It's a first druggable bucket, fully resolved for HIV. I think it's the final resolution is around 4.5 or 2. With the help of filters and with the help of good detectors, and you can try to visualize the features very clearly. And this is how the total workflow of tomography looks like. With the cell culture, you try to vitrify and try to identify your area of interest, and then you using an ion beam milling process, and later you try to go inside the holder and try to see the particle of interest at different angles, and then analyze the data with different softwares, Amida or other relevant softwares. This is one reference example from Julia's group. I would like to share this. So as a whole, I would like to summarize. So irrespective of single particle analysis or cryotomography, or else even if you take micro ED of interest, so the main important points, main important point behind it is how we are trying to answer a biological question of interest, and how we are trying to work on the purification protocol, and once the samples are optimized, which methods we are trying to which method we are trying to use it, and what is our ultimate objective. That should be clear. And with the advanced equipments and with the advanced filters, it's achievable. The high resolution is really achievable. Atomic resolution is achievable, and you can also try to the equipments are also compatible for different biological safety labs, BSL 1 to 3. And yeah, this is just I want to share as an example how quickly you can try to load the sample, and there is no need of microscopy expertise. You can try to learn around the technique and try to collect the data on your own actually. And this is a kind of an advanced software with a smart EPU. Even during the data collection process, you can also try to, the smart EPU can try to filter good and bad particles, and there you can get an initial result actually. Prior to the image processing software, there is a small analysis that smart EPU can do. And with this, I would like to conclude my presentation, and I hope you might receive a few insights regarding the NSTEM as a technique, cryo-electron microscopy as a technique with different particle types, and then later few insights into cryo-electron tomography. And I would like to touch upon micro-AD, but I will take that in the next session. Thanks a lot for listening to my presentation, and I again sincerely apologize for the technical glitch that we faced initially. Thank you. And if there is any questions regarding the, if there is any questions, please ask. Otherwise, I will try to on my camera.
Hi, everyone. Please, if you have questions, could shoot it now.
Well, let me begin by asking one question. That is, you said staining, even small changes in the staining agent used make big difference. Do we understand why such big differences occur?
Big differences in terms of contrast, or else in terms of particle orientation.
So if, for example, you talked about formate versus acetate, in a tungstate situation.
Mm-hmm.
So overall, the contrast is because of the tungsten and the electron density of that.
Mm-hmm.
How is this big difference occurring? Do we know this?
I think it's mainly to do with the properties of the staining agent. So probably with the phosphotungstic acid, what we can see is we can try to see. Of course, in all the three, there are a lot of staining artifacts, but the less artifacts, we can try to see visualize more information as possible. But upon using all the three, I would recommend the optimal concentration of uranyl acetate, at least 1 or 2%. Of course, it will affect your particle contrast, but you can at least see the top view of your particles clearly. But PTA or uranyl format, somehow I couldn't. I mean, I'm negative on that actually. But PTA, you can try to see side orientation. With less harm to the particles.
A related thought is that many of these metalloproteins, there is already a metal present there. And now, how do they behave with and without staining?
What kind of metals?
Metalloproteins, proteins containing a metal atom.
Like apoferritin is a metal, like.
Yes.
I think the stains, the interaction with the stain, probably I have to come back to that question, sir, to answer it in a clear way.
I mean, do you sort of altogether exclude staining agents when it comes to a metalloprotein?
No. I will come to that, sir. I will come to that. I just don't want to answer. I will go back and then research and then come back to that.
Okay. I leave it to you, Dr. Ritesh.
Okay, thank you. Yeah. So anyone else having a question?
Query like, let's say you have protein of like 66 kilodalton or 30 kilodalton.
Is it Subrata?
Yes, sir. Yes, sir.
Yeah, I mean, you can actually tell your name before you ask questions because nobody can see you, right?
Yeah. So this is Subrata. I'm working under Professor Pradeep only. So the what I'm asking is that, so let's say you have a protein like 30 kilodalton, 60 kilodalton, and 132 kilodalton. So let's say you have protein with different size, different molecular weight. So which kind of staining agent can be used for this one? So is there any kind of information available to you? Like, let's
So
say
you
high
mean
molecule.
the functionality of protein is monomer or dimer or trimer, or is it what is
No,
the
let
functionality?
yeah, functionality means all are like monomer. So one is like 40 kilodalton, one is
Oh, okay,
66.
okay.
Okay, so any means which kind of staining reagent can be used, like uranium formate acetate or tungstate, or any kind of
No,
information regarding that?
irrespective of different sizes,
Okay.
you can start with uranyl acetate as an example, actually, and you will see your particle of interest, irrespective of sizes. Doesn't matter actually with the stain. With uranyl acetate, at least you can see the features little bit clearly. At least you will get a rough idea of how your sample looks like. PTA, you will get a lot of artifacts, and also you will get disturbances. So you might have a difficulty in identifying your sample, but at least uranyl acetate, you will get a better clarity.
Yeah. Also, you have mentioned like 3 microliter sample for preparation of negative staining. Like 3 microliter is okay. What about the concentration? Like how much concentration means initial concentration we can go with for
Yes,
any kind of
your sample of interest. For example, if you take protein, I will tell in mg per ml,
okay.
approximately 0.001 mg per ml is needed. Um, and for cryo-electron microscopy, approximately 0.5 mg per ml is needed. At least 20 to 50 times more for cryo-electron microscopy. And if you prepare this concentration, you can try to dilute 10 to 20 times for the negative stain.
Yeah, okay. Okay. Thank you, sir.
Or even more dilution might be needed, because the higher
Yeah,
the dilution, you can see the sample dispersion quite clearly in negative stain.
okay.
Um, uh,
Yeah.
Shubhrata,
Thank you.
just to add on to that, although the concentrations you need at higher concentrations, at the end of the day or before doing the experiment, you might have to optimize to get a, a clean distribution of the proteins, which the softwares can easily identify during the particle picking stage. So you might have to do some optimization work before that imaging itself. Just wanted to add on that part. Sorry, Ram.
Yes.
Okay. Yeah.
There are alternative techniques, at least to measure the protein concentration, or else any other samples. But to get a rough idea of concentration, it's better to have a rough idea. Then based on that, you can try to dilute your samples for NSTEM.
Okay.
But before that,
Thank you,
if
sir.
you try to go without concentration information, if you try to prepare the samples, then I think you will be wasting a lot of time actually in the microscope.
Yeah.
Anyone else has question? I mean, for negative stain, there is one more thing that you all should know, that your protein should not be in phosphate buffer. That's a must. It should be any other buffer like heaps or tris, but phosphate should always be avoided. And when you are diluting, probably you can dilute it like 10-fold every time. And that generally gives you a range, and then you can work in that smaller range to get to the concentration that actually works for you. So, I mean, my question to Dr. Sapnappan is that when you work on MSP and when you try to build a nano disk, then you purify your MSP and then you add back the DOPC or DHPC, or you can already have kind of, I mean, what's actually the process of building these nano disks?
So there is a optimized ratio actually. So what's important over here is lipid to protein concentration.
So I mean, my question was, suppose someone wants to make a nanodisk of 50 nanometer, or someone wants to make it to 100 nanometer or 150 nanometer. So what actually regulates the size of these nanodisks when you have a constant of MSP, or do you change MSP in that case, or do you change DOPC or DHPC?
Yes. You can try to the MSP. So you can try to by different constructs with different sizes. Actually, it starts
so
from
MSP actually regulates the size of those nanodiscs, right?
Yes, exactly.
Okay, yeah, that's what.
So how long you can try to build a belt? It depends on the MSP constructs, and also it depends on the amount of lipids that you infuse. But anyhow, the lipids will be sorry. But anyhow, upon adding the detergent, most of your lipids will be removed, and it tries to form a belt-like structure.
Yeah.
So it mainly comes from the construct size.
So is it possible that after you have created these kind of nanodiscs, because obviously you cannot regulate the size so much, that then is it possible to size them and separate different sizes of nanodisk, or you have to use a mixture of that?
So initially, what happens is once you try to prepare a good stoichiometric ratio of lipid to protein ratio, you will get a crude mixture. So crude mixture meaning you will get empty discs, and you have to purify it. And upon purification, probably based on what we see.
What's the method of purification for this one?
I did it actually. There is a small step. I forgot the technique. I will mail you, Ritesh.
Okay, great. Yeah.
There is a small screening step that I usually do.
Sure. Yeah. Okay.
I will come back to that. Yes.
Sure. So, any more question? Please, this is a good time to shoot it.
Upon curiosity, is there anyone working on nanodisk here?
I mean, I wanted to work on nanodisk, but yeah, that's. I mean, it's actually quite a bit of work to work on different concepts of MSP, and I have few constructs, and well, it's actually available through AdGene. One can buy it directly, and then people can use a DOPC. So actually, DOPC and DHPC is something that you have to try with different kinds of MSP to get like various sizes of nanodisk. That's what is.
Yes. There is a optimization process that we have to do. Correct amount of lipid to protein ratio is important here.
I have one more question. Like, I mean, when you talk about the carbohydrates, especially those glycosylation sites being probed using cryo-EM, generally the like signal to noise ratio that is generated when there is a protein, or when there is a protein attached to RNA, or protein attached to DNA, they generally generate a little bit more contrast, especially RNA and DNA associated with the protein, right? Compared to protein alone. But when you're talking about only glycosylation sites, they generally have a very high, very low signal to noise ratio. Then how do you actually commit to different orientations? I mean, do you have to collect the tomography data as a to prove the concept, or you can just depend on SPA to be able to analyze their, to assign their
for the site specific data, I think it's better to collect different orientation first to get an idea of different angles, how the particle looks at different angles. Then based on that, I think even Spark can do that, but tomography in specific, it's for this purpose actually.
Because the data that you showed about the viruses, you are talking only about the cages that is with the protein, but when you talk about the very fine details that is actually associated with that, those are like glycosylations or carbohydrate residues that is like protruding out of it. Those structures are not going to that high resolution of 2 or 3 angstrom, right?
No, I think it's recent is 4 angstrom, something like that.
Along with the glycosylation or without glycosylation?
Without
glycosylation. Okay, yeah, that's what I wanted to know. Because people are still working on refining the structures beyond the cage that is with only the glycosylation. That's where the docking actually happens. Okay.
I think that's why tomography is slowly building up actually.
Yeah.
Because these are the kind of like probably limitations of SPA as well.
I had one more question. If no one else has a question, I can just go on. But otherwise, if you have a question, please shoot it now. Okay, so I just go ahead. You said that we use foam var support or carbon support for negative staining, right?
Mhm.
I have a very bad experience of having different kind of carbon creating different kind of background. I created my own grid with a normal carbon coating. It worked perfectly, all right,
Mhm.
and the foam var worked very good. But when I bought quantifoil carbon grid that has 2 nanometer carbon thickness support, and I put my sample on it and stain it with uranyl acetate or uranyl formate, it gives me so much of artifact. What could be the reason for that?
Probably the
Because it's
thickness of the
no,
carbon.
no, because it's actually quite good here, right? I mean, when I'm talking about only 2 nanometer thickness, that's like super thin. And I used it to gain more contrast for the particle and less noise coming from the carbon. But it did not generate any good image, even with apoferritin. So, I mean, the point is that, or for that matter, even we used very simple reference called catalase that works very well on any kind of carbon support for negative staining. But as soon as you put them on the quantifoil 2 nanometer carbon support, it gives very weird staining with the same stain. Do I mean, do you have such experience, or like, could you explain me why it could be?
I haven't used the 2 nanometer, but I can share my experience with different grids probably.
Yeah.
Back home in my lab, I used to prepare my own carbon film. We have a old evaporator over there. So when I tried to do like a faint carbon film and also like thick and thin with different thickness. But from my preparation, sometimes it is stable, sometimes it doesn't. So I think it depends on the support and how well it is supported on the grid. Probably that particular, I would say that particular supply is not good, but otherwise, I don't think there is no other reason that you can see actually.
Yeah, I mean,
But sometimes the carbon will be wiped off during the preparation. That's because if the carbon is too weak.
No. Actually, well, when we saw that under microscope, so there was carbon support. There was no problem with that.
Okay.
It's just that when we reached more than 9 nanometer thickness of carbon support, it gave me a very good image with the same sample. But when you go below, like something that is already prepared with 2 nanometer, it was a very weird result. Anyway, so don't worry about it. I don't think. Okay, someone else may also report
I used
something.
grids from Tetpella actually. It works good actually. Tetpella a little bit expensive, but it works. At least the batches works good.
Yeah. I mean, Tetpella means you're talking about C flat, right?
Yeah, C flat.
Okay, C flat. Okay, yeah, that's great. Okay, guys. So I hope no one is going to add any more question. Is it? I'm able to unmute myself. I think there is. I think there is a question in the chat. Could you look into that? There is. I think Sachin Mishra. I think he is talking. I'm unable to unmute myself. Some technical glitch. Can you ask a question for me? Actually, my protein shows precipitation in low salt concentration, so I'm using 250 millimolar salt for storage buffer. If I go to cryo-EM, will it affect the contrast? No, Sachin, actually 250 millimolar sometimes works pretty good. I would just recommend you not to freeze it. If you can go straight away, that's even better. So, Samyappan, if you can comment on it.
Yes, I agree with you. I think you will end up seeing a lot of crystals actually. The lower the salt concentration will be better for the cryo.
Yeah. I mean, lower is better, but if we cannot go beyond like below 250, sometimes 250 also works fine. It's okay. I mean, yeah, it's okay actually. Maybe you will see some kind of crystallization of ice somewhere, but they will. I mean, you will always find a spot where it can actually work. So it's fine, I think. I mean, you should just try, I think.
For KCL, yeah, both are fine.
Sure. Okay. So I don't think there is any more question that we are getting. So Dr. Samiyappan, thank you so much for giving us a good talk, and hope we can see you next time with another series that you talking about micro ED, right? In the next session.
Yeah, sure.
Okay.
Either me or one of my colleague will.
Sure. Yeah.
But I will like to thank this for providing me this opportunity, and also I think soon there will be cryo facility that will be coming up in your facility, right?
Yeah.
So
Yeah.
I will try to meet people as well. So let's keep in touch.
Yeah. Well, thank you so much, and thanks everyone for tuning in. We will sign off now, and you guys can grab your lunch now, I think. Okay. See you.
Yeah. Thank you. Bye-bye.
Bye. Yeah. Bye.
Thank you.